How does GDNF affect gene expression? Have you ever wondered how certain proteins can influence the health of our brain cells? One such protein is glial cell line-derived neurotrophic factor, commonly known as GDNF. This protein plays a vital role in the survival and function of dopamine-producing neurons, which are significantly impacted in Parkinson's disease. So, how exactly does GDNF affect gene expression? When GDNF binds to its specific receptors on neurons, it sets off a series of events inside the cell. This process is known as intracellular signaling. The binding activates pathways like the MAPE-ERK and pi 3 degrees Kelvin AKT pathways. These pathways are crucial because they lead to changes in the activity of transcription factors. Transcription factors are proteins that help control the expression of genes. Through this signaling, GDNF promotes the synthesis of proteins that are essential for neuronal health. For instance, it can enhance the production of dopamine, which is vital for movement control. GDNF also supports mechanisms that help repair neurons and promotes factors that prevent cell death. These effects are particularly important in the context of Parkinson's disease, where the loss of dopamine neurons leads to motor control issues. Research has shown that GDNF can rescue dopamine neurons and their connections in the brain, specifically in the nigrostriatal pathway. This pathway is crucial for regulating movement. The neuroprotective effects of GDNF are linked to the changes in gene expression it induces, which help maintain the function of dopamine neurons. However, it is important to regulate GDNF expression carefully. If GDNF is constantly overexpressed, it can lead to unwanted side effects. Researchers are developing innovative systems to control GDNF expression in the brain, ensuring it is produced only when necessary. This approach aims to improve safety and therapeutic outcomes for patients. Another interesting aspect is the role of nuclear receptor-related factor 1, known as Nur1. Nur1 is a transcription factor that interacts with GDNF and is essential for the maintenance of dopaminergic neurons. If Nur1 is deficient, it may hinder GDNF's ability to induce beneficial gene expression changes. This interaction could explain why some clinical trials using GDNF therapy for Parkinson's disease have not achieved their intended results. It is also noteworthy that increasing GDNF levels does not always guarantee improved survival of dopamine neurons. Studies with genetically modified mice have shown that altering GDNF levels does not significantly impact neuron survival in certain models of Parkinson's disease. This suggests that how GDNF is delivered and regulated is crucial for its effectiveness. In terms of treatment strategies, GDNF's ability to influence gene expression presents a promising avenue for neuroprotection in Parkinson's disease. Current research focuses on methods to deliver GDNF or regulate its expression in targeted areas of the brain. Gene therapy approaches, such as using viral vectors to deliver GDNF genes, have shown potential in promoting sustained expression and supporting neuronal health. In conclusion, GDNF affects gene expression by activating signaling pathways that regulate transcription factors responsible for neuronal survival and function. This mechanism is at the heart of its neuroprotective potential in Parkinson's disease. However, careful control of GDNF expression and its interaction with other factors like Nur1 is essential for achieving effective therapeutic outcomes. Always consult healthcare professionals for personalized medical advice regarding treatments and therapies.